Okay, so today we're going to talk about protein synthesis. There's two parts to this process. First one is transcription. Second one is translation. I want to show you a tip before we get started. So, oh no, sorry, I won't my pen. So these two words, they look so alike, right? Literally the first five letters are exactly the same. They both start with trans, all right? The next letter for transcription is a C. The next letter for translation is an S, or sorry, it's an L. It's L, right? So transcription with a C, and then lation with an L. In the alphabet, the letter C comes first, right? A, B, C, it's like the third letter. So that one comes first in the alphabet. That process happens first. This is a nice tip if you're on the test and you're like, oh my God, I can't remember. You're like, oh, okay, this one has C in it. C comes first in the alphabet. That one happens first. L comes much later in the alphabet, right? Way after C. And so that is the second process we're going to talk about today. So just tips as we get going. Okay, so this protein synthesis. I like to think of the analogy of baking a cake when I'm thinking about this process because I like to eat cake. Okay, and so protein synthesis is like when you have a recipe. All right, like when you're baking a cake, you start with a recipe. Maybe this is grandma's recipe. Okay, this is like the DNA. It is the written directions on like exactly how to make that delicious cake. Then I have to actually make it, right? I got to get all the ingredients together, stir it up, bake it in the oven. That is protein synthesis, all right? So this is making the cake. And then last, we get the cake, we get to eat it, right? At the end, we get a beautiful cake. I've never made a cake that looked like that. Maybe I need some lessons. But in the end, we get a beautiful cake. In the end of protein synthesis, we get a protein. So let's break apart this word. When we synthesize something, sorry, I need my markers. When we synthesize something, we are making it. All right, synthesis means to make. Here, we're making proteins. So it's called protein synthesis. That's what that word is when you break it down. And here we are using DNA as the recipe. All right, these are the directions for making, proteins. Here we're trying to make a protein. When you're making a cake, you're trying to make a delicious cake like that. All right, so step one, we need to copy the recipe. All right, so let's say, you see a recipe on the internet, right? We find like on blogs and all kinds of stuff. I find recipes on Pinterest. But then I want to make sure that I don't lose it. I take a screenshot. Love taking screenshots. Anybody else? You guys take screenshots with your phone? Well, I love screenshots. Okay. So you don't want to lose it. You take a screenshot of it, right? That's what we're doing here. We are making a copy of the recipe. So we've got it ready. Okay? So step one is copying the recipe, and this is called transcription. That's the one with the C. Remember? C happens first in the alphabet. Transcription, when we break down this word, I like to break down the words. Transcription has the word script in it. Transcription. Script means to write. Like when you see fancy script, or some people like do correct calligraphy, right? That's transcription, or writing something. So here we're going to write the recipe, and we're going to copy it. Why do we need a copy? Okay? The reason we need a copy is because DNA is kept in the nucleus, right? And it actually can't leave. It has to stay in there. It has to be protected. And it also, believe it or not, is too big of a molecule to escape. So even if it wanted to get out, it couldn't. All right? So we need to make a copy, or we need to take a screenshot so we can have it, when you need it. Like when you access it later in your photos, instead of having to dig through Pinterest or try to search it again in Google, right? So DNA, we keep it in the nucleus, so we need to copy the recipe. And the copy is called messenger RNA. And you might have heard about this, like the COVID-19 vaccine. It's a messenger RNA molecule. So we're going to learn about those today. It's an mRNA, that's the abbreviation for it, and it's a message. It's just a message, all right? For the COVID-19 vaccine, it's a message that tells your body how to fight the virus. It's a message. Here, we're taking a message or a copy of the DNA. So the DNA, it's stuck in the nucleus. We're going to make a copy or a message. And then in the end, remember, we're trying to make a protein. Protein synthesis is making proteins. Okay, so DNA, we've talked about this before, is a really long molecule, right? It's crazy long. And to keep it organized, to make sure that we don't have mistakes, because remember, mistakes can cause a lot of problems. We talked about that in our last unit with mitosis. To stay organized, the DNA is copied as codons, or it's copied 3 at a time. So 3 letters at a time, or 3 bases at a time. So for example, A, T, G, or C, if that's what the smartboard wants. This would be a codon, because it's 3. It's a triplet, okay? And so if you check out right here, I've got my molecule, and you can see I've got 3, G, C, and U. That's a codon. So it'll type it 3 at a time. It'll like copy it or organize it in 3s. So something interesting about RNA is RNA actually, you could see here it's got G, it's got C's, it's got A's, there's an A, but it actually does not have T's. So this would be like on your MacBook keyboard if the T was missing. And instead of the T, you had a U. So believe it or not, it has a different base that replaces T. It uses U, which stands for uracil. All right, uracil. So the way that you can tell if you're looking at a molecule and you're like, I don't know what this is, if it has U's in it, it's mRNA, or it's RNA, all right? RNA does not have any T's in it, thymine. It just doesn't have it. It doesn't have it on its keyboard. The key is been switched out for U. So we have some rules. This one's the same, right? C and G, they go together. We already know that. But here's the difference, all right? There is no T's. A matches with U every time in mRNA. So let's take a look at this picture here. Transcription is happening in this picture. So here we've got a piece of DNA. So here's the chromosome. I know it's kind of fuzzy. A piece of DNA. And we've got the DNA up here. I know that that's strands DNAs because I see T's. You see those T's there? So that's a piece of DNA. And here we've got this bar that's highlighted. That's mRNA. It's making a copy of the DNA molecule. Remember, messenger RNA is the copy. So here we're making a copy of the recipe. So step 1 was copying the recipe, right? We copied it. It's in an mRNA molecule. Now we need to bake the cake. Time to make that delicious chocolate cake. Or in the case of protein synthesis, to make the protein, right? This is called translation. Remember, translation has the L, and L happens second, or after C in the alphabet, right? All right, so translation is when we are going to follow the recipe to make the protein, like you would follow the recipe to make the cake. All right, and so I just want to remind you of this throwback. In the cells unit, we learned about ribosomes, and we drew them as these like little dots, right? We're like, yay, ribosomes, they're the dots in the cell, woohoo, and they make proteins. Well, here we are. We're circling back to it. Those little dots are actually really important. And we're going to talk about how they actually make the protein. So here we go. Step 1, the mRNA molecule, remember the copy of the recipe, is going to go to a ribosome. All right, there it goes to the ribosome. Each codon, oh, there's that word again. Remember codon 3? So that was the 3 bases, like C, C, G, right? 3 bases. So each codon codes for an anticodon, all right? So let's check out this graphic. I know it's really small on your screen, guys. I apologize. So here we've got the mRNA molecule, and we've got a codon. Remember, a codon is the 3 bases. Here, it's AUG, okay? As you might have noticed, these bases, they like to matchy-match, right? In the DNA molecule, it's the 2 bases. They come together. They make the ladder, right? In the transcription we just talked about, matchy-matchy, more matchy-matchy. Here we go. So our mRNA, the message, or the recipe, is going to match with something called a tRNA molecule. So this is another type of RNA, and it has an anticodon. You can see the 3 bases, right? And they're the opposite, matchy-matchy. So at the ribosome, we're going to have the mRNA come over, and then these tRNA molecules are going to come and match. They're going to match up with that message. This is like when you're bringing the eggs, you put them in the bowl, the flour, you bring the sugar. We're bringing all the ingredients to the ribosome. And then at the end of the day, right, we mix it all together, put it in the oven, we make a cake. Here, we're going to get a whole bunch of amino acids. Amino acids are attached. You can see they are attached to this tRNA molecule. And that's, again, these amino acids are like, here's some flour, here's some eggs, here's some sugar. So they're all combining. And in the end, we are going to get a protein. All right? So I know that was a lot of steps and a lot of like crazy chicken hand movements. We're going to watch an animation, so don't worry. If you were like, that was a lot, we're going to watch it in motion. You know, I like that. But before we do that, I want to hit a few more things or just summarize. So remember, first step, oh, I got to activate your marker, sorry. First step was transcription, right? Where we copy the recipe, right? This is the copying. Second step was actually making the protein, and that's what we get here in the end, right? That's what we're trying to get. So this is a 2-step process. We copy the recipe, bring it out into the cell, take it over to a ribosome, do translation, and make a protein. Okay, last thing, and then I promise we're going to do that animation. Okay, so the proteins. What is a protein? You are made of proteins. Your hair is made of protein. Your eyes, your muscles, like your entire body is made of proteins. So remember, each codon, I keep using this word, the codon is those 3 bases together. So like, for example, you know, C, C, G. You know, we've got 3 here. All right, so those are going to be a codon when we have 3 of the bases. Each one codes for an amino acid. All right, it's the code for the eggs, for the sugar, for the ingredients that you actually need. And so when we put all of the ingredients together, those ingredients are those amino acids, we get a protein, okay? And you are made of proteins, as I said, your whole body. This graphic really helped me understand it, or I felt like I was like, wow, this makes sense. So here I've got a piece, here I've got a chromosome. Remember those from our last unit. And then remember, chromosomes are made of DNA. So DNA is all like shoved into the chromosome. So here's a piece of DNA, all right? So this is like a gene. Okay, so here's a gene. This codes for a protein in the flower. That protein happens to be for a pigment or a color. So this protein makes that flower purple. So that's really just like how this works, is the DNA is the code for your hair color, for your eye color, for your height. And the proteins are what make you that height or hair color, stuff like that. So kind of an interesting process.